welcome you on behalf of Professor Bram Singh Horticulture Foundation, that is BSHF, a not-for-profit organization, and my own behalf to this 5H webinar series talk number 19. BSHF is thankful to NEPA, New India Publishing Agency, New Delhi for sponsoring this webinar series. I'm happy to welcome co-organizers Dr. Preetam Kalia, ICR Rafi Ahmed Kidwai Awardee, former head Division of Vegetable Science and coordinator School of Horticultural Sciences, ICR IRI New Delhi, and Dr. Shalendra Rajan, former director ICAR Central Institute of Subtropical Horticulture, Lucknow. Today, the webinar is on Fusarium wilt and the future of India subtropical banana cultivation by Dr. T. Damodar, Principal Scientist and Head ICR Soil Salinity Research Institute, Regional Station, Lucknow. Friends, banana you all know is important food, fiber, even the food plate crop in India. It grows in length and breadth of the country. Importance of management and control of most common disease, Fusarium wilt in banana in subtropics can hardly be overemphasized. Those who are in the banana production, they know the importance of this banana wilt or the banana Fusarium wilt. The disease outbreak takes heavy toll in the area of its spread, particularly subtropics. Lot of research has gone into it. The current status of disease, its management, prophylactics, and control measures, including future program on this disease and identical one, would be covered in today's talk. Friends, questions can be raised in comment or chat box. We will take up after the talk. Now I request co-organizer Dr. Preetam Kalia ji for formal introduction of the speaker, please.
Thank
Dr.
you, sir.
Kalia.
Thank you very much. Good morning, everyone. Most respected founder of VSHF and organizer of 5H Future Horticulture webinar series. Co-organizer Dr. Shalendra Rajanji, and today's speaker, none else than Dr. Damodran Tukaram. Dr. Damodran, who is going to speak on Fusarium wilt and the future of the India's subtropical banana industry, a very important topic in fruit science, especially in today's world. Dr. Damodran has had his master's and doctorate from Tamil Nadu Agriculture University, Coimbatore. He started his career as ARS scientist from Central Agriculture Research Institute, Port Blair, and rose to the position of senior scientist at Central Soil Salinity Research Institute of ICR Regional Research Station, Lucknow, where now he's serving as principal scientist and head. His research expertise in agriculture, horticulture science lies in splicing dynamic rhizosphere microbial diversity of perennial fruit crops for inducing abiotic and biotic stress tolerance in agri-horticultural crops to reduce the use of chemical fertilizers and fungicides that are of great concern to eco security. Dr. Damodaran the research contribution is addressing the outbreak of contagious banana Fusarium wilt disease in the Indian subcontinent through a holistic research approach of mapping the hotspots and developing a biofungicide ICR fusicont using a unique trichoderma isolate on a patent protected dynamic media. This product is currently being granted a global license for marketing internationally to address the devastation of disease in the Southeast and Latin American countries, apart from the Indian subcontinent under the Atmanirbhar Bharat program. Dr. Damodaran developed and patented an innovative in vitro bioimmunization product based on antifungal lipopolypeptide biomolecules and protocol for inducing disease tolerance in tissue culture banana plantlets at cell culture stage. Dr. Damodaran contributed immensely in exploring the unique polyamroni and wild mango biodiversity of Andaman and Nicobar Island and conservation of rich mango biodiversity of the Bay Islands. Dr. Damodaran was instrumental in adding nearly 66 polyembryony accessions of Sino-Indian origin to the National Repository for Rootstock Studies, and he developed a salt-tolerant polyembryony regular-bearing rootstock ML2, which was found to be better than the standard Israel rootstock 13-1. Dr. Damodaran also documented the genetic diversity of mango rootstocks using molecular markers for conserving the mango biodiversity of the country. And he contributed to increasing the productivity of horticulture crops in degraded and salt affected lands through the development of cost effective, eco friendly bioformulations using consortia of salt tolerant microbes. Dr. Damodaran also developed the next generation biosmart formulation, CSR Grosure, for enhancing and enabling production of fruits and vegetables in the soils of a higher pH up to 9.2. Grosure has been widely used in the vertical farming of vegetables as a source of potash and zinc solubilizer by some of the commercial growers of Bangalore. Dr. Damodaran is a recipient of several national awards like Jawaharlal Nehru Award for his work on banana resistance breeding, biotech product and process development, and commercialization award from Ministry of Science and Technology, and Young Scientist Award by Indian Science Congress for his pioneering work in horticulture and eco-safety initiatives in research. He is also the Fellow of Indian Academy of Horticulture Science and Society of Applied Biotechnology. Dr. Damodaran has published more than 70 research papers in high impact journals and has 4 published patents to his credit. With this significant contribution of Dr. Damodaran, the viewers are interested to listen to him on this very important topic, Fusarium wilt and the future of India's subtropical banana industry. So, not standing for a long time between the viewers and today's speakers, I invite Dr. Damodaran to take the floor and deliver his talk. Dr. Damodaran, please.
Thank you very much, sir. It's a very elaborate thing. I'll just. Respected, most respected Chairman, BSHF, sir, And I thank you very much for giving this opportunity to share our research findings and keep our thoughts for each special attention in the not only nationally, also globally also. I thank you very much, sir, Dr. Bram Singh Saab. I also thank Dr. Kalia Saab for making a very elaborate description about my work done. It's very grateful and I'm very happy to have this, sir. And I'm thankful to Dr. Rajan and other co-organizers who are there with me. And without wasting any time, with the permission of the host, I'm taking the right into the topic. So now we are going in for the most address some of the important aspects that subtropical banana industry is facing and will be facing, and what are some solutions and what are the solutions we have in our hand also. This is one of the important things. Before going into the deep subject of subtropical, let us know what is the difference between a tropical and a subtropical banana industry. The simple difference is subtropical industries where the bananas are having an additional stress of low temperatures also apart from tropical zone. You can see if you say our Indian subcontinent itself, the humid subtropical zones that starts from UP, Bihar, West Bengal and extends up to the northeastern, they have a typical low temperature regime compared to the tropical zone, which are very favorable for banana cultivation. And here, apart from this stress also, the another aspects we can say is the varieties are peculiar, and the only biggest stress the plant faces during the growth time is the low temperature, low winter temperatures. And that is why usually in subtropical zones, it was yearly very popular for from Bihar up to northeast. But UP, recently you can see how the banana industry is in UP has grown like anything. In 2012, it was just 2.35 thousand hectares, but whereas it gradually grown because the major reason is the intervention of G9 banana. From the history, the G9 banana, the Grand 9 cultivation, which is prone to have some tolerance for the low winter temperatures, has resulted in the increase, the drastic increase in the area. Now, as of 2017-18, the NHP data shows that it is 69.38, but the data of the state government of Uttar Pradesh, it reveals that it is 95,000 hectares in UP on 2021-22. That means it is coming next to Maharashtra in the area where it has crossed the bounds of other countries and traditional banana growers. And more 99% of the cultivation is under G9. And this is also a threat when a complete cultivation comes under a single crop and monocropping. Why this divergence has taken place in this part of the country, which is also a largest state in in our Indian subcontinent? The change has occurred because of the economic remunerations. People have started diverting from the rice and wheat system and even from sugarcane system to the banana cultivation because of good yield, good economic returns, and the water is surplus here. The one of the major reasons where when we compare to the southern part of the India and other traditional belts like Maharashtra, Gujarat, where they have used the water very preciously and it's a precious resource. Here also it's a precious resource, but the groundwater table is higher and the states are used to have regular monsoons, flooding, and usually the more than irrigated conditions, rain-fed water is that is water source from the canals. They also help us in help the banana industry. That is the reason for the change. Now, when the change is there, when the area has increased, the challenges are also increased. And what is this Fusarium wilt? Why this subtropical banana industry is peculiar to this Fusarium wilt? You see, Fusarium wilt is a very common disease that has been the major cause for the decline in the banana industry since 18th and 19th century, we can say. The most favorable banana variety, Gros Michel, as was nearing about that is extinct completely from the global scenario just because of this disease, Panama wilt disease. It's famously called as Panama wilt disease because its outbreak was larger in the Latin American countries. So that's where the journey starts for the Fusarium wilt, and it was race 1 we designated because it has a lot of races. Race 2 attacks the culinary types of banana, race 3 the ornamental types. So there were race 1, race 2, race 3, and race 1 was very devastating. It ruined the entire banana industry. The economy of the Latin American countries and South Asian countries relied on this banana industry. Once this disease has come, the Latin American countries were in a big problem and the entire plantations were devastated. But when there was a problem, there is used to be some solution where the superhuman power, God, supernatural powers disposes us, and that's a outcome was in Kew Garden. There was one plant that survived for this race one banana, and that was today Cavendish groups of banana. A lot of Cavendish groups came. It was like Robusta, Harichal, that is called as Harichal, then Dwarf Cavendish, Giant Cavendish. A lot of improvements over Cavendish claims, and people, as the changes came, the quality also enhanced, but the shelf life and other things was also challenged. But G9 was one such variety which had two major advantages: that a very high yielding variety which serves the economics stability, and second one is it has a that is thick wind, so that the shelf life is also increased comparative to the harichal and other things where immediate softening occurs. So this attracted the growers. This attracted the growers for entering into the banana business in the non-traditional areas of Uttar Pradesh. Then Bihar is known to have traditionally nearly 34,000 hectares are there in Bihar continuously over under the cultivation of this thing. But as you cross the borders of Uttar Pradesh and go to Bihar and West Bengal, apart from G9, the cultivation also depends on the very traditional cultivation cultivar, that is Malbog, which is famously called, and Martaman, called in that is in West Bengal. So as you go to the northeast, like to the when you enter the Assam, Malbog is one of the richest cultivar. You can see the biggest Asian market for Malbog lies in the that is your Assam Golpara district, that is Darangiri banana market. This is one of the biggest market in Asia where a lot of Malbog, and the rates goes up to 80 rupees per kg also. That is the value of this fruit at. And as you go through Tripura and other Tripura, the same Malbog with some changes due to the ecological change, eco factors, it is very delicious fruit comes from them. That is a shabri. It is one of the traditional and delicious fruits of Tripura. But unfortunately, this Malbog and shabri varieties were also prone to the race one of the banana. And what is this race one, and what is when we are going to race one, we are going to see the race one. This race one is such a dreadful disease, though it will not attack the Cavendish bananas, that is this G9 and other thing, but it is attacking and our traditional cultivars, it is called as Amrutpani in Andhra, and Rastali in Tamil Nadu, and Malbog in West Bengal, Martaman in West Bengal, and Malbog in Bihar, Assam, and other northeastern states of like Nagaland, Impal, and Shabri in your Tripura region. So this race one, slowly the inoculum builded. And the thing is, there is no replacement. If for Malbog and Shabri and other plantlets, only your sucker plantations were going on monocropping and different types of planting system. In Assam, it used to be in huge areas under Malbog, whereas in Tripura and other parts, it's grown in the homesteads. Such is the history where the race one slowly inoculum build up, and now it is posing a dangerous danger. Okay, now we are constantly, where people started working and addressing for the Malbog. So what is the status of this other G9 cultivar, which was so long been resistant or tolerance to this race one of banana? That's why I'm discussing the race one of banana. This is G9 is tolerant. That's why this Cavendish group came. And but as our pathologists, great pathologists like Plewitz, who have the entire career on the Fusarium wilt of banana, they predicted long before the continuous cultivation of G9 or the Cavendish groups of cultivar may result in one day a mutant of race one, which will be more dreadful, like how we called your normal COVID and Delta variant. Like that, and another variant may come in that, and that will be more dreadful than the earlier race one. So we have to be prepared. They started warning light from the early 19 and 1910 and something, and the research was going on, but to their warning, the situation came. Slowly, the race one perished in the, that is the Cavendish bananas that have been cultivated in the Australian regions and this South African regions. There, they started witnessing the subtropical race food. There was the incidence of Fusarium wilt was once again noticed in the so-called tolerant variety of the banana, that is your Cavendish groups. Then people started working on it, and it was restricted only to the subtropical zone. So they call it as STR4, that is subtropical race 4. But it was not too far. In 1990, a new variant of race 4 was once again witnessed in Taiwan and Indonesia, and that variant were much more virulent than your initial race 1 variant and subtropical race 4. And this tropical race 4 doesn't see any difference in any conditions or any varieties. It completely devastates all the things. right from the tropical and to the subtropical. But now the question comes, why you are focusing only on the subtropical banana industry and leaving the tropical banana industry? The comparative threat concerned with the Indian subcontinent is that the comparative threat from race 4 is lesser than the your subtropical zone, because when we analyze the entire data of climatic scenario, myself and Dr. Rajan, we were modeling the entire climatic scenario where all the tropical race 4 has been a very devastating thing. There, like Jordan, Lebanon, Bangladesh, Pakistan, Australia, everything we consider, there the unique feature was wherever the temperature goes less than 10 degree for a period of 2 to 3 months, there the tropical race force incidence is very higher, and the vulnerability, we can say the vulnerability index is high for these zones rather than where there is semi-arid condition and the temperature range is very minimum, varies from 22 to 40 to 35 degrees Celsius. There the risk is little comparatively lesser. And to our fortune, the tropical race 4 till now has not been reported in any part of the southern traditional belts of the banana Cavendish cultivar. Now the question comes, why we should bother? Why the subtropical people of India should bother about this tropical race 4? Yes, because it didn't left India till 2018. We were of the thing that we didn't had any tropical race 4 incidents, but unfortunately, the tropical race 4 in the year 2019 was witnessed in the Sohawal block of Ayodhya district in Uttar Pradesh. But years before, 1 year before that also, there were very sparse reports of the incidence of the wilt in Bihar Katiyar region. But to that much, we can say intensity, it was not spreading. But when the news came from the Sohawal thing, we were taken, and along with our we can say our lead institute, our National Research Center for Banana from Tamil Nadu, they were very kind and we collaborated and we went there, and to our astonishment, entire devastation was there. To be very, I can say very frank to friends, that on the month of June, when we visited the Sohawal, the disease incidence like this type of orchards, there were only 2 to 3 we can say plantations were there, and we thought we should do the intensive thing, because before saying it is a tropical race 4 or it is a banana Fusarium wilt, because it's a very tricky disease and identification itself is very complex, because the symptomology of this disease coincides with lot of other diseases like collar rot or the bacterial wilt, which is caused by Erwinia. And second thing is your nematode damage, that is your your weevil damage, the same symptoms are there. And the most similar symptoms are also witnessed in the subtropical zones where there is a specific pinter, calcium deficiency is there. And calcium deficiency also gives this thing. So what are the unique symptoms to identify it, which we adopted? One of this, the yellowing starts from the mature leaves and progresses towards the younger leaves, whereas it is a reverse in the case of your weevil attack. It starts from the center core and comes to the thing. So this was the thing, and sometime in any leaves it can affect. And second thing, your calcium deficiency and your Fusarium wilt, the yellowing will be very thick yellowing, and it spreads like how we are drawing in the diagram, like that it spreads over the leaves. So this is one of the symptoms, but yellowing cannot be alone the same single symptom to take it. And another thing, how can we differentiate from the collar rot, which is having the same symptom of yellowing and pseudostem splitting, which is another important feature, that is the pseudostem splitting. But in collar rot, what happens, mostly it affects the plantation within the 5 months after planting. But Fusarium wilt starts its expression after the 5th month, and more virulent expression comes 7th and 12th month. So that means you can imagine, if a plantation, if the symptoms are going to get expressed and devastating during the 12th month, the entire money the farmers have invested in the plantation comes to zero once the plant starts dying. And as I said, in the June, there was only 3 plantation. In August, the entire village was affected. It was spreading like a rapid fire. So the entire government system was called. We were addressing meetings. We were called by our honorable DG at that time. And then immediately our colleagues from that is NRCP, they suggested awareness is the important thing, and identification of the tropical race 4, that is very important before declaring it's going to be the race 4. So we started working on that. So how do we plan our work? Because it was spreading like anything, and by that time when we were doing, most of the districts were affected, and we came to know that the disease is spreading like anything. So first thing is we faced our way of approach, that is community sensitization, because once the pathogen comes, it remains for 30 to 40 years in the soil, and it is highly transmissible by soil, water. And in Indian conditions, we can see where the flooding type of irrigation is being followed in this part of subtropical zone, there how the spread will be. And even a tractor that has been plowed inside or used in the infected field, if it goes to the uninfected field without proper care and attention, or even the footwear of the farmer, if it goes to from one infected field to another infected field, it is going to affect the plantation, and it has affected. So community sensitization was considered to be the one of the important tools. We joined hands with all the NGOs and other people, and we started first immediately to sensitize the problem farmer. That is because it's very difficult to say to an Indian farmer, you don't go to this field, it's having a Fusarium wilt, you don't use the chapel, you don't use your tractor, you have to wash your tractor with T-Pol. All this, if you say, it was very annoying to them. But on seeing the intensity of the disease, the people understood, because we identified in 2019, but there was disease. Based on survey, we came to know the same place, the disease was slowly growing since 5 years. They thought it is a collar rot. They were procuring some insecticide from fungicides from Bayer and other Excel and other thing, and they were dumping it on the plantation. No result. But the inoculum slowly built it up, and it came as a bursting, devastating manner. So the second step comes as identification of hotspots. Without knowing where are these hotspots, we can't face our control measure or your containment measure, how you are going to go. And third thing is the immediate solution, because there are only three solutions. One is you should go for a tolerant variety, which is a permanent solution, but it takes years to come. And your second is your awareness that you're already doing it, so that you can prevent the disease spread from one field to another field. And third most thing was biological control. Biological control is the immediate measure and a tool. But if biological control would have been worked, since 90, this has been creating a big havoc in the South Asian countries like Taiwan, Australia, China, Japan, everything. By this time, they could have control. No, it is not at all possible, because this is a perennial crop. In an annual crop, if you are going to apply a biocontrol agent, the results will be visible, and very short duration, your results are going to come. 3-4 months, your results will be visible. But it's a perennial crop, and you have to continuously target the phenological stage of the crop and also the incidence pattern of the disease. Then only we can define the role of the antagonist. So thing is true assessment of the efficacy, because we are going to play with the fungus, which is challenging one, and not any babiotic stress where yield can be reduced or anywhere the yield can be had. But it's not like that. It's going to be completely reduced. So the pot culture, clinical experimentation and evaluation of pot and feed culture with the virulent strains. And the fifth phase is that once you're formulating the antigen and large-scale testing, as I told, community-enabled adoption. Because in a country like India, only community-enabled adoption can do it. I will show it in the later slides. So this is a thing we just came up with a very clear idea. We are going to survey and map. This is a prime important thing. So we have to map it. And second thing is, once we are mapping, we should identify. That is also very important. Identification of isolation and identification of race 4 of Fusarium wilt is really a challenging task. I should thank our NRCB scientists. They helped us initially, so we could take up in a very large manner, and we could do it hands in hands with them. So identification and confirmation. And third comes the management. Once it is confirmed, we have to go in for the different steps of management. Today, I will be dealing with all these 4 things, how we progressed. It's a journey we progressed, we can say. So you can see in the first thing is the UP. In Uttar Pradesh, these are the regions that are completely reliant. That is Devariya, Maharajganj, rise from the borders of Nepal. The borders of Nepal, the Narayani River enters from here into the thing, Maharajganj. From here, intensive banana cultivation is being stepped, because surplus water, canal water is there, no depth of water, and a very favorable summer temperature, that is your March to June, the very favorable climate, and huge market for this thing, because usually the whole UP used to get bananas from the Bhusawal area. So there is a huge market when it is generated here itself. So these entire districts, you can see these are the banana growing districts. These green districts are still not infected, but these are the thing on our survey we came to know all these districts are completely affected. More than 40% incidents were there in this thing, and it was spreading like anything. Initially we identified the disease here, and as we went there along the canal zone, the complete areas, both the sides of the canal were being found to be infected with this disease. Huge plantation loss, farmers were lying, but then also they were relying on banana on the hope, with a ray of hope that this disease will go and we will get the income as usual. Some farmers used to say, we have been minting money like anything in this banana, we don't want to lose this industry of subtropical banana. So we started working on that. Similarly in Bihar, we are fortunate because they have a blend of G9 and also the Malbo cultivation. These areas are that is your Katiyar, Bagalpur and Purnia, these three were intensively affected, and all these three districts completely nearly to 20,000 hectares are under the, out of 31,000 to 34,000 hectares, 21,000 hectares are under the cultivation of G9, and the rest of this 12,000 to 14,000 hectares in Vaishali and this Vaishali belt, there they go for Malbog, Gazipur place, where Malbog, the race 1 is severe, but not as severe as the race 4. Even the disease reached Sitamarhi also, which is very far away from these places. How it reached, I will just discuss in the next slide. Similarly, as we grow to the other subtropical zone of your Assam, which is well known for the Malbog industry. Now the Malbog industry is coming down. In markets, the Malbog industry is coming down. Slowly, Chini Champa is coming, even though of low market value compared to Malbog, because there are no other alternatives. Large areas in the both the sites of in the banks of Brahmaputra, they grow this banana, especially this Malbog cultivars. You can see these three districts, Golpara, Kamrup and Barpeta. These are the highly infected districts with the Malbo, that is Panama wilt, that is a race 1. Here it is race 1, and in Bihar and UP it is race 4 was very prominent. So these, but here the race 1 has grown up, where the loss is nearly 30 to 40% in these three districts. And large big growers are also there, FPOs are there, who are nearly waiting to a solution to this. Such is the case in the Assam. When we come to Tripura, these three districts, that is Safajila, West Tripura and Koval, these three are of this complete, this Tripura comes under the Shabri cultivation, which is one of the very, very famous cultivar of this Tripura region. And this is also with under the threat of race 1. And now these three districts are completely being affected with this race 1 of this Fusarium wilt in the Tripura region. And similarly, not only with this, we have even surveyed Nagaland, Impal, everywhere the race 1 is a problem. But in these two states, there is a real severe problem of this Panama wilt. So you can see the symptoms, friends. You can see how the plants, bearing plants, are dying. This is in the parts of Sant Kabir Nagar, Gorakhpur, Sohaval, and this is Kaptan Ganj in UP, and how the plants are falling at the bearing stage. And this is the typical symptom, I can say. When you decipher the plants, you can see the complete vascular discoloration. This is one of the important symptoms of this Panama wilt diagnosis and morphologically. And next comes, similarly in the race 1 also we surveyed, and here there is a little difference. The intensity is not that as much severe as it is in your G9, where tropical race 4 is there, and we are lucky enough that your tropical race 4 has not reached the northeastern states. Still they are in the clutches of the race 1, which can be managed. And we also should take some of the policy initiatives, which we'll be discussing in the current coming slides. How can we prevent these states from the threat of this Fusarium race 4 also? So then there comes the identification. As I told, it's very difficult to identify, and there are initially people who are working on molecular markers with tough markers. Then specific markers, Dita et al, they developed, it's a scar-based markers, that is 463 base pairs. But when we started screening, it could not differentiate between a race 1 and a race 4 also. The race 1 was also amplified when we use the data marker. That means the reliability is reduced. So we thought, and another Lin et al, they also developed one more marker based on the scar markers, but that was also very staggered, because when we collected samples of race 1 from Assam and the southern parts of the country and compared with the race 4 what we have collected also, they also showed irregular, not repeatability was not observed. But gene-based markers, which was developed based on the secreted xylem cells, and these gene-based markers could 90% it showed reliability in differentiating between the race 4 and race 1. And we can say that nowadays, as of today, the best marker to differentiate between a race 1 and race 4 is the markers developed by our NRCP colleagues. It's wonderfully working. We are validated in wide range of cultivars, because we have to correlate the symptoms of phenological symptom, intensity of disease spread, and the molecular identification. That is why I say it will be very difficult for a person or a very good pathologist to differentiate between race 1 and race 4 in banana and subtropical race 4. And we should thank these people, and they have come up with excellent markers, and it is working very good. Now, up next stage, what we should go, I will just discuss in the last when we come to the strategy slides. So apart from that, we even check whether the authenticity of the markers by using some ISSR unique markers also, to be very clear. This complete Bihar and UP, where the isolates were collected, they were coming under the race 4, and other parts where we have collected the disease plants also, even from southern India, like Coimbatore, we collected from Chennai, we collected from Gujarat here, and all they're showing amplification for race 1 only and not for race 4 under both the 6-1 markers and also the NRCB markers. So we also did some next stage studies like BCG studies also, but this is the way we confirm and this is the way we progress. Now comes, okay, we have identified what we are going to do. Say we have to see how this disease is spreading. Unless you don't know how it is going to spread in this subcontinent, you cannot control in the subtropics. So we found, because most of the TC banana requirements of this UP, Bihar and other places met from your southern tissue culture laboratories, where they send the primary hardening and a secondary hardening nurseries are a source of industry in the UP and Bihar as a source of remuneration also. That is 45 days and distribute to the farmers. But here you can see how they take the water from the canal where the disease plants have been thrown. This is one of the mode of spread. That's why I told you, from UP, from one of the nursery from the UP, the material has gone through Sitamarhi, and where there was no district in Sitamarhi, the disease is being witnessed in a very wide range, because the nursery has also attributed the hardening nurseries. And most of the hardening nurseries use the water from this software infected source they use karte. And another thing is your flooding, because you can see these are all lowlands where continuously subjected to flooding. The flood waters, you can't control the flood water from one field to another. You can control the irrigation water by bunting and other thing. When it is flood and the changing climate changing scenario here, where concentrated rainfall is being witnessed and floods are very common here, and it is a big challenge now to control the spread of the disease through flood waters. So with all these constraints, how we are progressing, what are the solutions? I have given a very grave picture, and there is also green side of this. Let us go to the green side now. And we started working, as I told, from the biological control measures. Most of the isolates of trichoderma we were having, we collected from the suppressive soils, and fortunately that is the science. What happened was, the isolate that are being collected from a highly sodic atmosphere where the pH of the soil was 9.4, that started performing again. That's why the science of antioxidants and stress tolerance, which is physiological phenomenon of each individual microbes or human being, whatever it is, it started proving. That is why in a selection we should not restrict only to just simple biology, that is your disease means only biotic areas we have to collect. We have to concentrate on wider area of stress adaptability, so that we can get wide diversity, and that can be useful. To our astonishment, we got one of a very novel strain, Trichoderma resi, that showed nearly 83 to 85% in vitro control compared to other Trichoderma. So we started working on it. We took it to the pot studies. The second stage is the pot study, and based on the pot, this is where rhizosphere, from banana rhizosphere to Trichoderma resi. Trichoderma resi is a very unique and very different from other trichoderma. Most commonly, Trichoderma arzianum is being used, Trichoderma kuningiapsis is being used, or Trichoderma asperellum. These are all the three viridi and Trichoderma viridi. These are all the four trichodermas that have been extensively used in the biological. Where this resi come? This resi is very unique and it is very difficult to culture also. Resi is very unique and very difficult to culture, and it has high cellulolytic and proteolytic property. That is why it is being used in the food industry. It has been first time isolated from the German scientists during the Hitler's regime to degrade the military bags that were accumulated after the Second World War. That was the invention it was made. That is the property of cellulolytic degradation. But fortunately, we were able to get the resi here, and it performed very beautifully against the controlling this disease. So we went for it showed very clinical control over the disease, and now more than pot field evaluation is very important. We took it in the hotspots regions. We had an intensive field efficacy because it's a challenge. We have to, and field application means we have to schedule the time of application according to the disease expression and according to the phenology of the plant. And that is why subtropical climate ke liye the scheduling will be different, whereas for a tropical climate, the scheduling of biocontrol aspects is going to be different because of the variability of expression of diseases in this region. And based on the positive results in the field experiments, we came upon the product. That is why this product is unique. It is a simple biofungicide. It is obtained using Trichoderma resi. It's one of a unique stage that has shown clinical control over your tropical race 4. And what is the uniqueness for the sustained disease control? Because biological agents are not famous for giving a sustained disease control. What is the reason? We developed a specific media for that. Specific media and such a thing across the pH, the formulation can work. If the formulation can work in acid pH in Assam region for race 1, and whereas little higher pH in like near alkaline pH to our even to our that is endogenetic belts also, and even normal pH of southern tracts. So these are the things we devised, and we developed a specific media. We got it patented, and we got it registered, under the NBA approval also for using the mixtures which we are using in the patent. The patent is an accordant patent media, and this is the combination of a dynamic substrate, dynamic media, and a dynamic isolate. These three has resulted in deciphering the mycelium of the Fusarium because of the earlier growth, fast, rapid regeneration, and occupying the entire space before the Fusarium can go into sporulation. These spores are formed and they tear the mycelium of the your target FOC. And what are the need? Once a product is developed, it has to go under a lot of regulatory clearance to come into the commercial stage. We can't just take it to 2 huge hectares and for commercialization. So thanks to our council or SMD Horticulture, they are kind enough to support, and our DG ICR, seeing the results in the farmers field, they supported it to go in for complete regulatory clearance. So we went it for bioefficacy data with multi-location trials. We went it for generation of toxicological data because we are handling a biological product. It should not have any toxicology to human, to aquaculture and the ecosystem. And third is the shelf life studies, because this is where each and every product fails. And our product sustains because of the large shelf life and proven shelf life. That is very important to maintain the optimum load of the fungi there. And packaging studies, this is also very important. All these studies we conducted, and the product was registered with 93, with a 93B license from the government of India for domestic and global markets. And based on these results, Down to Earth conducted a third-party evaluation in the fields where this was a disease were there to assess, because it's a big disease other countries cannot address, how India is claiming they have addressed. So they came to the fields, they interacted with the farmers, and third-party evaluation were done, and they made it publicly publicized in the Fresh Plaza in South Asian, which is a South Asian issue. And based on that, a lot of global demand was there. People are demanding from Japan, people were demanding from Philippines that we want to use it. And luckily, here MS Innoterra India is a Swiss company, along with collaborated with Desi Fruits of India. They evaluated the product in their fields for 1 year, and they took it for license, for global license, and production will be in India under Atmanirbhar scheme, and it can be marketed globally. So now in 7 places that they are taking trials in Philippines also using this Fusicon formulation. And for domestic need, Kandelwal Biofertilizers has taken, total ICR has got 45 lakhs just by licensing, and we are getting royalty also. 35 lakhs is going to come to us as royalty, because 1 lakh hectares people have started. The government has given subsidy for this program. So thus is the impact, and the wonderful product. It is performing excellently. Now, can we sit quiet because this Fusicon has come? It is going to perform, because we can't sustain on the biocontrol control measures over a long period of time, because the pathogen also has very intelligent, and it can produce mutants. Before it produces mutants, we should be very clear to restrict the use of this Fusicon to 2 or 3 space instead of 4 to 5 space, which we are recommending drenching, so that it can have a sustained thing. So we started deciphering what exactly is happening by application of this Fusicon into the plant system, how it is attributing tolerance, plant, microbe, and fungi, that is pathogen. All the 3 we started studying the interaction. We started deciphering the metabolitic studies through LCMS and GCMS studies. Key metabolites we identified very unique to the Trichoderma risi, and also in the plants which has been drenched with this Trichoderma risi, we could come out with unique lipopolypeptides and antifungal compounds, that is iturin C, phenigizin, and peptabiols. It's one of the important products that has been released by the Trichoderma. And xyloglucan. Xyloglucan, it is a glucanase. It triggers the glucanase enzymes that breaks the cell wall of the your pathogen. And Soya sapochogenol is the highest form of your antioxidant that gets expressed only on stresses, when the plant is exposed to stress. So all these were unique in your Trichoderma risi, and they were also found to be unique. That means science suggests that we have to go further. we have to exploit something. So what are the genes it triggers? We compared it with the fungal toxins genes also, which is been associated with the Fusarium. And wonderful result we got it, to be very because when we presented in the global media, they were asking, this RISI is very much prone for your known to be non-virulent, and it does not have any antagonistic property. How it is controlling your Fusarium disease? It is usually used in the food industry to break the carbohydrates and proteins. And we started deciphering the science. Yes, it is having two genes very prominent in it, CBH1 and CBH2. CBH means that is cellobiohydrolase. CBH2 is very common under all the circumstances where EC used to exhibit. But CBH1 is expressed only when a fungal or a biotic stress is being introduced into the plant system. Wherever it goes, if the resides into the system, then it expresses the CBH1, that is a cellobiohydrolase, which gets expressed to its maximum only on the induction of the stress. And this has paved the way to devise the mechanism how our bioagent is controlling. Based on this mechanism, we started thinking, if we are applying a material as a drenching to a plant on the fight from the 3rd month, 5th month, 7th month, 9th month, why can't we take this material into the plant system? Because banana, fortunately, it's grown by tissue culture plants. So tissue culture, how can we address this problem? Then we came, there are two theories. One is this normal biohardening, existing biohardening theory is from shooting, you go for rooting. From rooting, you go for primary hardening and secondary hardening, whatever it is. In primary hardening, your bioagents are added here by drenching. So that we call it as a biohardening in the nursery stage itself, and it goes to the planting. But there you cannot, since soil is such a macro media, you can't control and say that whatever I'm spraying is going into the plant system and it is going to come till the end of my growth when the fungus is going to attack the thing. So we had some failure. That is why there is sporadic results when you go for biohardening coupled with Fusicon. What we thought was, why can't we study this? So we started working on these protein molecules, selective filtration, membrane filtration. How we can isolate some of these important biochemical that is your lipopolypeptides, and screen off all the impurities in the system. Because as such, if you are going to add any biomolecule based, fungal based biomolecules to the tissue culture media, what happens? It induces mutation, it induces more phenolic content, so that the survival rate will be hampered. So we were very clinical, and that's why we patented the entire protocol, how much we are going to use it in the shooting to rooting media, and at what time and what concentration this polypeptide molecules are going to be integrated into the plant system. Once we had standardized, and excellent results we got it. And the plants we obtained from these immunization, that is, we call it as bioimmunization, because it's using the biomolecules of the and lipopolypeptides that are obtained from the Trichoderma rhizae. So whatever this is, and we also blended with some of the antifungal compounds also. So once we went it for a typical pot experiments without addition of your Fusicon or anything, and the plants showed excellent rooting, and the disease was controlled completely. It was not at all expressing, that is a race 4. Then we took it in a larger scale. Unless we evaluate in a that is in the hotspot regions, we can't conclude it. So we went it into the larger scale, and nearly in the first year we evaluated 35,000 plants in both the hotspots of UP and Bihar, that is Katia region in Bihar, and Sohaval and Maharajganj region in the UP, where the disease is in a very we can say vibrant manner. We evaluated, and we had two such of evaluations, only with immunized alone, and immunized with just 2 times supplementation instead of 4 times or 5 times supplementations of Fusicon in this area. And when Fusicon alone is applied, the disease control varied from 83 to 84 to 90%. Mostly it comes to 83 to 85, the average disease control. But when we blended with this tolerant immunization technology, it was very astonished. It got up to the 94% also. We have got most of the plantations were sustained, you can say. Completely the fruits we are having very good yield in that. So both the sectors we had documented the results, and in the second we took it we applied the patent, we got the patent for this, and last year we evaluated 4 lakh plantlets of this, not only in G9 but also for race 1 we did it with Malbok and Shabri also we multiplied, and that also we are evaluated. And for 4 lakh plantlets of this G9 through incubative model, we had some of the budding we took them as an incubator under partial commercialization scheme, and we entered and we supplied the material at the rate of 12,000 rupees per liter. Per liter can sustain 20,000 plants they can grow in per liter of our material. We charge them 12,000 rupees for this thing plus GST. And we have last year it went up to 4 lakh plantlets we have developed using this technology. And this year we are having requirements for more than 100 liters for this material, such because the results are very good. 2 years data has been completely documented, and we start. That's why Fusicon, when supplemented with this bioimmunized plants, is going to be a very good solution for the burning problem of tropical race 4 and race 1 also. And DBT joined hands. Seeing these results, DBT volunteered us. DBT took us for funding, and we started working in 2 other institutes. That is Assam Agriculture University and our Noida, that is Institute of Horticulture Technology, where they have their institute at Mandira in Assam. They also joined hands with us, and from Agriculture College from Tripura. All these people we are joined together, and CASH and ourselves, we joined together, and large scale. Now we took it because most of this, what was happening was the entire Marburg industry was coming down, and slowly G9 was starting peeping in, because in Marburg the race 1 is affecting the trees. So people have started shifting, and there was no tissue culture planters available in Assam and Tripura for these Marburg and Shapri varieties. So they thought, why to grow for a monocropping with the sucker-based population? Let us divert to the G9 where we are getting. But on our venture, we made FPOs. Through FPOs, we created FPOs. Through FPOs, nearly now we have 1,200 acres under our control in both Tripura, especially in Golpara and Barpeta district. We have 2 FPOs, and 1 FPO from Tripura we have formed, and 2 farmers organization. We took them in control, and Fusicon based disease control measures, along with slowly we are replacing the old orchards which have been infected with FOC with TC plantlets. This year we have supplied 60,000 plants to both these states. And apart from that, the third thing, as I have told, the third thing is our NRCP. They are coming up with very good evaluation, and they also come up with some of the varieties. And we were also doing continuous screening in the hotspot regions, whether we can find any one plant which can show a tolerance, so that we can evaluate it completely. And when we started surveying right from Malda up to Siliguri, crossing all Bihar, all the districts of Bihar where the banana is being grown, and then up to West Bengal, we went there. We found in one patch of Siliguri where the disease, in entire patch where disease affected by race 1 and race 4, whereas yeah, 1 or 2 trees and the suckers which have been planted from those mother plants to other places in the neighboring orchards, they did not develop any disease at all. Then we collected the material, and we thought we'll go for a evaluation against race 4 and evaluation against race 1. We did it for race 4 in our UP condition for this Marburg 1 collection. We made it, and for race 1 we did it at in collaboration with AAU, Assam Agriculture University, Jorhat, and we also roped in one of our ACRP partners at that is Kovur, HRS Kovur, because they have one of a very good IMTP, International MUSA testing program, wilt sick plot, which has been maintained for more than 25 years. And any plant which is very difficult to survive, susceptible plant of Malbog or Mataman or your Rastali or Amrutpani, they can't sustain more than 3 to 4 months there. It is highly infected with your Fusarium and collarwort. So we evaluated in that, we gave them, this is the sick plot results of the first year, which they had shared with me. Dr. Snehilata, she is doing it. And without application of Fusicon, the plant is able to sustain. That means now we are going close to a source, but it needs validation of another 2 more years, so that we can come to a clear-cut indication how this material. But if this material plus your immunization technology and your Fusicon, whatever is available currently with us, can surely take care of this wilt in the subtropical region. That's why also these areas are the green areas. Initially I told the gray areas, and then I told highlighted the green areas. So what are the strategies for management? This is very important, because we should come up with the artificially intelligence based diagnostics. As we have done, we have done for drone based mapping. Dr. Rajan is here with us. He is very, very intelligent to taking us, and we have devised the methodology for identification in our surveys. We use drones for identification and also to assess the impact of the material, because every field we cannot go and assess. So drones were very useful, and AI technologies are very important, artificial intelligence and diagnostics, not only PCR based diagnostics. We are in need of LAMP based techniques, where non-PCR based techniques need to be developed. And currently in India, Japan companies are approaching us. They want to test, they have developed a diagnostic kit, they want to develop in India, they want to validate in India. But our partners from NBAM, National Bureau of that agricultural important microorganism, one Dr. Ilal is there. We are working with them, and he has come up with a diagnostic kit without using a PCR also, but it needs validation. We have to join hands and validate. Then second comes the management. Awareness is very important so that we can check the proliferation of the disease to different parts or different districts where it is not available. And regulatory approval and regulate, that is government support for already available products. Biocontrol, we have a Fusicon, and some of the products are also, it may be coming up. We have to support them, and we have to take it into the system of cultivation, into the banana cultivation, so that wherever it is affected or not affected also, if we are going to take, then it will serve as a prophylactic effect to control the disease in the initial stage itself. And TC firms that are supplying material to the hotspot districts has to be regulated through the government system. They have to necessarily take the in vitro bioimmunization, adopt the technology till another alternative is available for them, and take it. It is one of the method to have an prophylactic planting, that is healthy plant material cultivation, and this has to be done. And that, like the tolerant varieties research has to be still rigorous, come up with a very good tolerant variety. And varietal change, because mono variety, that is G9 and very good work, replacement of the G9 in the Theni district has been going on with red banana. People have done it from our colleagues, and it is one of a very important thing that will change the pathogen affected rate also. So we are also trying to increase the area under Marburg and other things where tolerance is there, and slowly adopt all the other technologies. So for doing this, there is a need for strong state support, and the state of Assam, the state of Tripura, the state of Impal, they are very kind enough, and the state of Parabani also, that is Northeastern Institute of Parabani, they are very kind enough to support these technologies, and they are taking it, and DBT is supporting. ICR, we are joining hands together, not only CASH, we should join hands across the institutes, across the things, and we can come up with a complete measure, and we can dictate to the world, globe. That's why we say community groups. Community groups were the success for our technology dissemination. Before the product got commercialized, we got it validated in 120 acres. That means your confidence levels goes high. And based on these results, now international consultancy, Philippines, Dole Asia Holdings have approached the ICR for giving a consultancy for immunization technology and this Fusicon technology. And apart from the Nipogene from Japan, they are also approaching us. And here is the where clutch we have, that our ICR can dictate. We can, if you're coming up, if you're working together, we can come up and we can find a holistic solution for all these, for a never-ending challenge, but a viable solutions we can find for this. So they were the team members who were associated, the very vibrant team. And Dr. Muthukumar is a biotechnologist, Nitish is a pathologist, he was working on diagnostics. Dr. Papi Bora from Assam Agriculture University, she has made a lot of FPOs and also isolated a lot of strains and taking the Fusicon technology and immunization plants. Kavita Yadav, she had a PhD in that race 4, and he is a person who took it to Bihar. And Dr. Sukandas is associated from Tripura, and Snehlata Rani, she is associated from your Andhra, and Dr. Manish from tissue culture sector. So these are the 4 institutes we have worked for addressing the subtropical banana industry. Friends, subtropical banana industry is growing, and future, the major banana can be produced from this subtropical zone if you are very vigilant and adopt very good measures and work with collaborations. Collaborations is the key for success of any technology that are to come, and we will stay together. Thanks a lot for giving an opportunity to express our experiences and teamwork in this lecture. Thank you very much, sir. Yes, Madam
With
Pinky.
a great hope to
Yes, sir.
get out
Yes, sir.
of it and be a winner only and have a successful banana cultivation, whether it is a hot spot or the less spread areas or disease-free areas, whatever there. You have covered almost all aspects of that. I'm happy the type of research you have conducted and the level of research, see, of a class, and which has been recognized by others also, and I'm sure such type of research will always be welcome, not only in India but abroad too. So now I request my colleague Dr. Shailendra Rajanji to add value to his talk briefly.
Thank you very much, sir. This was a very comprehensive talk on the Fusarium wilt and the future of subtropical banana cultivation. And he has very well introduced how the banana industry will be important, particularly in subtropics, and it is crossing many of the states, particularly in production. But also, he has given the reasons, economics, and other reasons why it is becoming. And he has also alarmed that how the nature of user build is causing due to tropical race 4, it is creating problem in Cavendish group in subtropics, mainly in subtropics. And he beautifully traced out systematically how the work was done on tropical race 4 in particularly in UP and Bihar, how the globally this disease is important and how successful the team was in managing it in India. Then he has shown the interesting journey of the success story, how the inter-institute collaboration in ICR institute collaboration and private partners and the farmers, how they can work hand in hand. And this is the success story which has been praised at many forums. Then he has given details of the UP and Bihar epidemics, particularly related with the G9, and also detailed how the Malbhog and Shabri is also being affected by race 1 and in Assam and Tripura, and successfully they are managing race 1 also with the technology developed for tropical race 4. The interesting way of identification of the and establishing how the tropical race 4 has played role in these both the important states UP and Bihar, and how to differentiate between the race 1 and race 4. He has given the details and why this disease is spreading in UP and Bihar. What is the mode? Why it is becoming so popular? This in an epidemic form. He has given that. He has also flagged the policy decisions to be made related with the this tissue culture material from the areas where the disease is existing. Then a story of the development of the ICR Physicon technology in a very, very interesting way he has given, and how Trichoderma raceyi has come to the rescue for the developing the technology against this disease. The management practice package was developed by the team where the ICR Physicon and immunization technology has is being combined, and how it is working. It is important for the sustainability of the management process. Then commercialization has yielded 93B license, which is honored for the technology, and it has increased the global demand, particularly the demand from Philippines and Japan. More in the technology has been demonstrated by ICR Physicon in more than 1 lakh area, but not particularly for mango, sorry, for banana, but also for cumin also. Then effort has been made for by him to make it the technology sustainable under the environmental conditions of the tropics, and the science behind the ICR Physicon and it how it will be furthered, the results, how those are useful, and what is the genetic basis for this technology. He has given the details. How the tissue culture plant immunization was possible through peptides, and the results were how they were patented in the in vitro immunization technology for in vitro immunization technology. All those details have been beautifully presented by him, and he has given also some details about the DBT project, how the DBT has recognized this technology and has supported the successful saving of the this for the successful saving of the Sabri and Malvo in Northeast India. Then details of the progress made in the tolerant or resistant types in Malvo tested in Tamil Nadu and Jorat. Those are also important, and strategies, and he stressed among the strategies he stressed on the community-based management of the for the higher success in this disease. He has given the entirely different presentation, which is in a holistic way. This was important for the subtropical banana, but also for the future management of deadly disease like tropical race 4 in of Fusarium. In conclusion, he has given a great lecture, and as a learned and experienced speaker in this area for the development of the subtropical banana industry. Thank you very much.
Thank you, sir.
आवाज नहीं आ रही सर? Unmute, sir. Please unmute. Sir, please unmute.
Unmuted.
Yes, yes, yes.
Could you kindly just add to it whatever briefly?
Sir, as Dr. Rajan has explained that we have had wonderful talk because Dr. Rajan was also associated with this program. And I think I need to say in this way that this is a very systematic study that they have conducted and gone step by step ahead in developing technologies and shown how the technology, I mean, it's a real depiction of the scientific that other sectors of agriculture who comments on horticulture, that horticulture people are not scientific. What else you need? This is the most scientific talk that we have had, and how this science has progressed systematically, because the science we do systematically is systematic study we do is the science. So that has been demonstrated by Dr. Damodaran here very much, and from the management point of view, diagnostics point of view, survey identification, every step has been taken systematically, but my which has been already detailed by Dr. Rajan. But I want and I'm interested most in the areas that key metabolites that they have worked on. So it's wonderful to know about actually how it works. What are the things which are providing, what is happening in the plant system, which is actually providing that sort of tolerance to the plant, and identifying different species of trichoderma. So I think there is a future in gene for gene hypothesis sort of study in this case, where fungus and host plant relationship has to be taken up. So that way we can progress further in that program to identify the genes, identify more as the tolerant varieties. MB1, they have already come forward, and they have demonstrated developing 4 lakh plants, as Dr. Damodaran has mentioned, and also shown that spread of the technology to 340 hectares has gone. So that is a systematic I mean, program which has taken forward, and which is helping farmers in containing the disease. And I think there is a lot of appreciation which are required for this type of work which has been carried out and which is benefiting farmers. So congratulations, Dr. Damodaran, and his team for a nice study conducted and beautifully uh, portrayed in the form of lecture today. So I think the viewers must have been benefited to a greater extent. Thank you very much.
Thank you, sir.
Thank you, Dr. Kalia. There are a lot of appreciation, but no question. Why the question is not there? Because the talk was very clear. He
Yeah.
started scientifically and ended with science. So
Yes.
those who understand the science, they have properly
It's okay.
understood his talk. And I further emphasize, science has the answer to our problems.
Yes.
Kindly respect the science and use the science to solve the problem. He has clearly, step by step, everything has brought out in this race how it could be successfully overcome. And he has given the future also know how to go about so that it doesn't create havoc. It may appear, but will not be able to create havoc. And ultimately, as Dr. Kalia has said, Dr. Uh, Rajan also has said, see, genetic constitution of a whether it is a host or whether it is a pest, both are important. So you have to mismatch. these guys. So, so somehow or the other, with the help of science, we have to just activate the resistant gene against that pathogen or insect or whatever the undesirable thing is there. We have to just activate them and to make them functional. Be aware, something may happen. And on the contrary, also, we can play with the pathogen also, can incapacitate the incapacitate the uh, insect, pest, or pathogen so that it doesn't become virulent on crop. Because we are selfish. We want to save our crops. We may be cruel to these pathogens or the pest because they also need food. They have to survive. But human, I think, on this earth is the most selfish creature for the benefit of himself or herself only. So you can't deny it because we have learned it from our God. So the God also taught it like that. So we have to be. So for that, the science, which I think I'm very happy with his way he has delivered it, the way he has conducted the experiment, and the approach, fantastic approach. I congratulate Dr. T. Damodaran, his team, and those who are associated with that, whether it is NRC Banana, whether it is Assam Agriculture University, or some other institute, and his team members particularly, I congratulate them. Each one of you have contributed, help each other. That is why the success. And I think, as I say, nothing is impossible if we work sincerely. The problem are there, but they are born with this solution. And those solution we have to find out. That's our job. And that's what I think the message from Dr. T. Damodaran. Banana is important. Banana has to face the disease. Banana has to face the deadly disease also. We have to protect them. We have to work from the very beginning so that we get the commercial crop. We are benefited only. We are not at a loss and we don't lose our important germplasm. You see, from his talk, the hidden message is there. You are concentrating on G9, only one genotype. So, and the ancient banana things, we have to maintain them, promote them. So, likewise, we have to do it. So, with these words, I profusely thank Dr. T. Damodaran for extraordinary talk. Then I thank to Dr. Rajan for nicely just moderating it, and Dr. Kalia for doing the rest of the job since introduction to that moderation and always encouraging, always helping, always assisting. And he see that if Brahm Singh has slipped somewhere, he will immediately just point out. He will point out before time and able with his help to correct it. Today also there was something, some glitch was there every time. Nothing can be smooth on its own. Every talk will be smooth if the preparations in the beginning are done sincerely with the good intention. That's what Dr. Kalia's thing. Thank you, Dr. Kalia, for taking care of everything. I'm thankful to Miss Pinky, who has helped us today. uh, Miss Shivangi, who was helping, she got married yesterday only now. So congratulations to her also. That is why
Sure.
she is not with us. I'm thankful to my daughter, Dr. Rachana, who is always there to support me if something is required in webinar, always inquiring about also, no? All the time, all the help. And I'm thankful to the audience. Without audience, the webinar doesn't have much of a this one. So I'm thankful to all the viewers and I'm thankful to one and all. The next webinar, we may be excused because of not one region but more than one region, we are not able to have it on 4th. We will have the next webinar and we will shortly inform you the topic, flyer, etc., everything. as usual. So allow us some sort of a rest or some sort of a chutti on 4th. Thank you
Yes, sir.
so much. And
Thank you, sir.
bye.
Thank you.
See you
Thank you.
again. Thank you.
Thank you very much, sir.
Thank you.
Thank you very much, sir.